So the problem we're addressing has to do with drugs and the way they interact with receptors in our bodies. What we are interested in is the class of receptors called the opioid receptors. These respond to opiates, which can cause pain inhibition in our body, and these receptors are actually present on the cell surface, and they need to be there to interact with these drugs. And if they're not present there, they can't interact with these drugs. And so what we're trying to look at is how the receptors are getting to the surface and what keeps them there or takes them away. One interesting opiate receptor that we are looking at is called the delta opioid receptor. This receptor is actually held inside the cell and not available to bind to drugs on the surface. So what I'm trying to study is a way to improve the trafficking of these receptors to the cell surface to allow these drugs to bind and activate the receptors for pain inhibition. In our lab, we take advantage of high-resolution fluorescence microscopy to visualize the movement of these receptors in live cells in real time. All of these receptors start their life in the endoplasmic reticulum. This is the factory of the cell, and the receptors are made here and they need to move on to their final destination. The first place that they reach after this is the Golgi apparatus, and this is the shipping and receiving center for the receptors. Here they can get modifications and have different sort of labels put onto them that'll help them reach where they ultimately need to be. What I've been able to determine so far is the mechanism that controls the intracellular pull of the delta opioid receptor and the mechanism that we can use to induce its trafficking to the surface. And we were able to work with collaborators to determine that when we did this manipulation and induced more trafficking of these receptors to the surface, we could actually inhibit pain in animals that had chronic pain that were previously unable to be treated. Instead of making new drugs, we can use old drugs but improve their response by putting more receptors on the cell surface. Being at CMU to conduct this research has been invaluable to my experience. The expertise in fluorescence microscopy with the EMBIC Imaging Center and the help from the Pittsburgh Center for Pain Research has really helped me to complete this project and probably would not have been successful otherwise. All of these experiences have provided me with the tools and resources necessary to complete this project and to prepare me for my future career in science.